Good. Hello and welcome. For those of you who are repeat offenders, we're happy to see you here. And for all the newbies, welcome. Once you come here, you're a member, and then you're free to invite other people. Again, remember our honor code, which is we only want people with good hearts and good minds and chock full of curiosity. So we say you come here with curiosity and you go away satisfied. Today, again, as always, I want to thank the Sanford Burnham Prebys Medical Discovery Institute for giving us this auditorium and the IT support and helping us along. I want to thank Doreen Schoenbrunn, our co-program director, and Rebecca Wright, our event manager, and my husband, who's not here today, who keeps insisting we have to keep doing this. So, and to all of you for continuing to be part of an absolutely lovely community, full of wonderful, caring people. And so many of you have helped us with programs and suggestions. So if we appreciate it, keep the good suggestions coming. My name is Rena Horowitz, and I'm the co-founder of this institute, of Group of 12, and my co-founder was the co-founder of this institute. Also, I would like to just make a couple of announcements of upcoming events because we like to share community. So on the 7th, there's going to be a Saved in America dinner. Saved in America, what they're doing is they feel children should be saved, not sold. And so they have an event at Estancia on Thursday. If anybody is still interested, they have a few seats there. I'll tell you how to get a hold of them. On the 10th of November, there's going to be a gala called Vision of Children, and it's exactly that. It's to benefit children who have extreme vision problems. And it's going to be at the Del Mar Marriott on the 10th. And the featured performers, for some of you who are old enough, are the Temptations. So that should be a really good event. Today, as I was thinking about doing this, I was thinking how lucky we are that we all live here and that we have all these famous, nationally acclaimed institutes on the Mesa. We have Sanford Burnham Prebys, we have Salk, we have Scripps, we have JCBI, we have the Institute for Immunology, and we have the Institute for Tropical Diseases. And from time to time, we're so lucky because not only do we have brilliant scientists from these institutes, but sometimes we have the heads of the institutes. So it gives us a chance to hear cutting-edge research and discoveries before almost anybody else in the country. So I think that's very exciting and it's pleasurable for us. And in the next couple of months, I'm going to announce something that's really big time and national, and we're going to hear the results from space before even NASA hears it. So that'll be kind of fun. And before I go farther, I would like to ask Joe Vecchio to come down and spend 1 minute because we have something new that today is the first time we're doing it.
Well, I've been bugging Rena for how many years have you been doing this, Rena?
You have only been bugging me for 2 years.
How many years has it been? 8, 9 years? 15 years. So we've had so many great programs here over the years, mayors and all kinds of celebrities and luminaries. I've been bugging Rena to videotape these in case we can't get here, we could see the program later. So finally I found an organization called Options for All. They're funded by the state of California, so it's no dollars out of our pockets. And they came here and checked us out. Somebody's got directions coming over. Oh, by the way, turn off your cell phones. I always bug you about that. In any case, you'll see 2 cameras here and a sound man and a crew manager here from Options for All. They are a nonprofit, but from now on we're going to videotape these courtesy of their organization. We'll put a YouTube channel up. If you can't get here, we'll be able to find the lecture in case you missed it. Okay?
So, you know, today I was thinking about our program, and I was thinking about how I was going to express what we're going to learn today. And it's about the mouth and the oral microbiome. And a thought came into my head because I was looking for old adages about the mouth. And so then the first one that came into my head was, it's better to be silent and be thoughtful than to speak and remove all doubt. But I decided not to go with that. So today, the adage is, you've heard the expression that you can hear it from the horse's mouth. So today, it's not the horse's mouth, but it's a beautiful, brilliant scientist, and she's going to spend some time telling you about your own mouth. And to introduce her, I'd like to introduce first Jill Mullen from the J. Craig Venter Institute, and she will tell you all about our speaker, Dr. Anna Edlund.
Thank you, Rena. I hope I don't prove true what your first line that you decided not to use, but you are all in for a treat. As Rena said, I have the pleasure of working with Anna and many other scientists at the J. Craig Venter Institute, but she's pretty special, so might be one of my favorites, whatever that means. So Anna is an assistant professor at the J. Craig Venter Institute, and she works in the genomic medicine group. She's also an adjunct assistant professor in the Department of Pediatrics at the School of Medicine at UCSD. She grew up in Sweden and obtained her PhD in microbiology at the Swedish University of Agricultural Sciences in Uppsala. She also has experience as a postdoc in natural products discovery, microbial chemical ecology, and human microbiome research. She's passionate about understanding how the human microbiome impact our health and how we can harness its metabolic and genetic capability to prevent and treat disease. And we will hear much more about that today. Dr. Edlund's research is funded mostly by the NIH and also through the generosity of private donations to the J. Craig Venter Institute. According to Google Scholar, Anna has published 43 papers or participated in 43 papers and has been cited over 1,500 times. Pretty impressive. And on the personal front, she's married to a charming man, Chris. She has a daughter, Madison, here, who is 15 years old, and she has another daughter who lives in Sweden. And remarkably, when you look at her, she is actually just became a grandmother. So here's Anna.
Thank you very much, Rena, Jill, Rebecca, for inviting me. This is a pleasure meeting all of you. And yes, so how are your microbiomes today? Any queasy feeling in the stomach or the throat? I have a little bit of a cold going on, so we should not do any hugging. So, anyways, today's seminar then is called The Human Microbiota, Our Gardens Within. I like to think of our microbiomes as our gardens that we should nurture and take care of. But it's difficult to do that because we don't know much about our microbes. We know that the human genome consists of 20,000 genes, which is an impressive number, but the microbiota in our bodies consists of 30 million genes. So we're actually more microbial than human, but we know almost nothing about the microbiota. That's pretty fascinating. So for that reason, I was going to give you, or I am going to give you a crash course, whether you want it or not, in microbiology, a very short one. And then secondly, I will talk about the microbiota in disease, and I will not only touch on the oral cavity, but also about the gut, because that concerns us all a lot. And then talk about the microbiota in health. And also I will lastly mention about what my research group is working on at JCVI to prevent and treat infectious disease. Crash course starting now. So I will tell you. I don't have a cursor like a laser pointer, so I will use this cursor here on the screen. Can you see it?
Yeah.
Cool. So to the left you have a typical animal cell, and to the right here you have a plant cell. On the bottom, a bacterial cell. What I want you to focus on is the highly structured internal environment of the animal cell and the plant cell. They have organelles. For example, the animal and plant cell have a nucleus. Inside the nucleus, there's DNA that is highly organized in a very specific manner. The DNA is packed together into our chromosomes. We also have other organelles, such as the Golgi that transports protein in a very particular way, and we have mitochondria. These are the blue little spheres here. And I want you to remember mitochondria, because I'm going to talk about them a little bit later too. So mitochondria, they are the power plants in our cells. They produce all the energy that we need. Plants have them too, but plants have plastids and chloroplasts that also provide energy to plant cells. Looking into the bacterial cell, it's very unstructured. As you can see, there's no organelles. The DNA is sort of floating around. It's maybe attaching to the interior, but mostly it's floating around. No organelles. Bacteria can have flagella, these little tails here. They can swim around, and that's very common for pathogens. They can actually swim pretty quick. Some bacteria don't have flagella, they're just non-motile, meaning they just sit still. It's very beneficial to be a single cellular organism because you can interact immediately with your environment. You can perceive signals and immediately respond. Multicellular organisms cannot do that. So bacteria, they can sample the environment really quickly. So they can, for example, pick up DNA that's floating around in the environment, and they can sample that DNA and they can keep it if they like it, as the case with antimicrobial resistance genes. So bacteria are very tricky and very smart. Looking at life from a different perspective and how all organisms belong together, this is the tree of life. This is based on phylogenetic context and evolution. So life started about 3.5 billion years ago, and we were all the same. Then it decided to take two directions, one into the domain of bacteria, which you have on this branch of this tree, and then the second branch split up further twice into a domain we call archaea, and the domain that is eukarya, which is plants, animals, protista, and fungi. So is there anybody in here who never heard about the archaea? Yes. It's pretty interesting because they're everywhere. We don't know about them yet. So they were discovered really late in 1980s, and they were found in hot vents, in black smokers in the oceans. They were found in all these extreme environments, but we have also found them in our guts, in our mouth, and they're very prevalent actually. And the reason to why we find them in these hot vents is that their membrane structures are very unique and hardy compared to the bacteria. They're also single cellular. So the archaea are very good in degrading small carbon compounds and produce methane from those. So if you happen to have methane production going on in your gut or in your mouth, it's the archaea, and that's actually a good sign. So something really cool, at least I think so, happened 1.5 billion years ago. It's called endosymbiosis. And this is basically setting the stage for life, more complex life, forms of life. So it started with that we had this primordial soup where primitive eukaryotic cells were existing together with bacteria. So this primitive eukaryotic cell, you see here, this little yellow circle, decided to engulf an alpha proteobacterium. Normally, the primitive cells would have digested it and used it as food, but this time it decided it was just going to keep it around. And it turned out to be a very beneficial interaction, which we call symbiosis. And it stayed like that for many, many billions of years. So the alpha proteobacterium in the end didn't need many of its genes, so it let go most of its genes because the host could provide all the amino acids that it needed for synthesis of proteins, and it could provide all the other vitamins and nutrients. So in the end, it had dropped almost all of its genes. And it turned out today in our cell, this is our mitochondrion, the one that I just spoke about on the other side. So, if I sequence this little mitochondrial genome, it will match up with alpha proteobacterium. So, it's pretty amazing. Another detailed view of life is this tree. I'm not going to bore you with more trees, I promise. So I'm very fascinated with phylogeny and evolution. So in this part of this tree, you see this is very branchy and that's all the bacteria that we have found so far. It's a huge diversity, and they're very dissimilar phylogenetically. So continuing down this path of the tree, here we find the eukaryotes, the plants or cells in this little green branch. So we're not so diverse. And here are the archaea. They're not so diverse either. So then we can look at this tree and we see that we have all these purple branches and with little red dots next to the branches. And that means that these bacteria have never been cultivated in the research laboratory. We have only found them by sequencing technology. So we've never seen them. We haven't been able to play around with them in the lab and look at what are they doing. However, they contribute to almost 50% of all the diversity in our body, in soil, in marine environment. And this is, of course, very exciting as a microbiologist to be a part of trying to understand what this diversity is doing. It turns out, when we look closer into this, that these bacteria in this purple area, they all have reduced their genome, just like the mitochondria. They have very small genome sizes. So, for example, we managed to isolate one of these bacteria in our research lab at JCVI. We call it Saccharibacteria, or TM7. It turns out it only has 700 genes left. Normally, a bacterial genome is about 4,000, 5,000 genes. This one only has 700 genes, and it has no genes for synthesis of amino acids, so it cannot build its own proteins. We're very curious when we identified this genome. And it turns out it's a symbiont of another organism. And it's the same for this TM6 bacterium. We also found the genome in a hospital sink, in a local hospital here in La Jolla, because we're going around sequencing everywhere. And it turns out we could find in the genome little imprints that were similar to an amoeba. And an amoeba is this single cellular animal that lives in waterborne systems. So when you cultivate the amoeba in the lab, you find that the TM6 live inside the amoeba. So then, to study TM6, you actually have to grow the amoeba in the lab. So, more about this TM7 that actually comes from the human body. We identified it growing in the lab only together with a host, this long, slender bacterium, an actinobacteria. And TM7 are these little round balls, you can see here, circle spheres. They're 200 nanometers in diameter, and they refuse to grow without a host. So they attach to the host, and they also regulate the growth of the host. So we realized that when the host becomes too abundant in the growth cultures in the lab, TM7 starts preying on it, starts eating on it to regulate its population size. Then the population, the actina, goes down, and then TM7 is happy, it stops preying on it, and everything is fine. So the actinobacteria and TM7, they were isolated from the human mouth, from biofilms growing on our teeth, which we also call dental plaque. We know a lot about the taxonomy in the human body, meaning that we know which bacteria are where because of sequencing technology and because that microbiologists have cultivated microbes from the human body for many, many years now. So we can look at different body cavities and we see that the taxonomy of bacteria and archaea and also fungi is very different in the gut compared to skin. The vaginal area has a completely different microbiota than other areas, and if we look, for example, inside the oral cavity, we realize that there's different microbes growing on our tongue as compared to the soft tissues and dental plaque and the biofilms on teeth. So we've been very good at determining what these microbes are, or who they are, but what we don't know is what are they doing. So I wanted to mention a really nice study that I appreciate from my colleagues at UCSD, Dr. Peter Dorstein and Rob Knight. They were sort of like a lot of people had the same thought at the same time as usual. We're all going after these microbial functions, we call it. What are they doing? So one way to look at that is to study the molecules, the chemistry that the microbiota is producing, because the chemistry can tell us whether the bacteria are responding to the pathogens. For example, they secrete antimicrobial compounds if there's a pathogen around that protect us from invasion. So in the figure to the left, there's two figures here, one to the left and one to the right. So there were 2 adult subjects involved in this study, and they were studying human skin. So they did these 3D maps of these bodies, and they swabbed each little spot that you see here. It's a cotton swab. They swabbed thousands of spots on these people. And first they determined the bacterial taxonomy on different body sites, and they also determined how many bacteria are where. And they found, for example, that hands, as you can see here, harbored a lot of different bacterial diversity. But if you look at this person's feet, there was almost none. It's blue. There were no bacteria on the feet. We don't know why. So then they did this overlapping chemistry profile for the same body sites, and then you could see what compounds are produced by the microbiota. So for example, in this figure here of the face, you can see that there's zones that are highly red. It turns out that these highly red zones contain oleic acid, and that's the acid, the oil that keeps our skin moist and healthy and protective barrier from invasion of the pathogens. And the producer of this oleic acid was Propionibacterium. So they could see the oleic acid in the skin on the face, on the neck, and also on the chest. So this is showing a very neat way on how we can map the chemistry and the microbiota onto our body and learn more what the microbiota is doing. So the microbiota has co-evolved with a human host, and it has changed its composition dramatically at least 2 times. So how do we know this? Because usually it's soft tissue. Microbiota grows as biofilms, or there's no skeleton. How can we know how the microbiota has changed over the last 15,000 of years? So then if we return to the oral cavity, it's pretty fascinating because we can see that biofilms growing on our teeth actually calcifies just like hard tissue, like bone, like shells, like plates. And we can find that dental plaque then fossilizes. See where my cursor is. So if you follow this red arrow, you see these little plaque areas. So we can collect them and sequence them. And the matches we get is the microbiota that lived in the oral cavity of this person. For example, here, a hunter-gatherer who lived 15,000 years ago in northern Africa. Then we can collect information like this from different time points and compare how did the microbiota change in different parts of the world in relationship to what diet was available. So then we know that human microbiota changed drastically from when we settled from being hunter-gatherers to become farmers, and this happened in Europe about 3,000 to 5,000 years ago. Because we got access to food containing a lot of grains and carbohydrates. It was a softer diet, and our microbiota then was selected towards degrading carbohydrates. And what happened with our jaws is that our jaw also became smaller. So we got problems with teeth crowding, which we still have today. A lot of us need braces. And teeth crowding is a problem because it supports development of periodontal disease, gum inflammation, and also cavities. And other problems that occurred when we settled and became farmer was that our jawbone got more porous. So when we were hunter-gatherers, we ate roots and berries and leaves, which demanded a lot more chewing. The second time our microbiota changed was at the introduction of the Industrial Revolution in 1850s approximately. And now I'm talking European terms, of course. The Western diet, like we call it, which is the highly processed flour and sugar diet amongst other ingredients, have been introduced to the world in different eras, but in Europe it started in 1850 approximately. In parallel, we see a dramatic increase in oral diseases such as periodontal diseases, that is inflammatory gum diseases and cavities. And we can compare them to our skeletal evidence from hunter-gatherers, where only up to 5% have had cavities, and today almost all of us have had cavities. So oral conditions are usually underestimated, and people may not go to the dentist. They just put it on tomorrow or next month because, yeah, it's just a little cavity or it's just a little ache, but it could actually result in pretty dramatic conditions if over time, that is. So we talk a lot about the systemic axis today in science, meaning that, for example, the microbiota oral systemic axis, meaning that the microbes in our mouth affect and impact other areas of our body. So, for example, in patients who have Alzheimer's disease or diseases related to cognitive decline, in some cases we see oral pathogens in those areas of the brain, and we don't really know if they cause the disease, and science is really dissecting into these matters right now, but we definitely see associations. And we know that in patients with different heart diseases, atherosclerosis, we find oral pathogens in heart plaque. Type 2 diabetes is tightly linked to gum inflammation, and that's a chronic systemic inflammation. Arthritis, preterm birth of babies, is also linked to oral pathogens. And the determinants of these diseases are major are Western diet, poor oral hygiene, we are not brushing, we're not flossing enough. A more known system is the microbiota gut-brain axis. And a lot of attention this area has caught a lot of attention in media, I know, so it's all about the gut feeling, like at least I still hear a lot about the gut feeling, and the gut is right, because the gut makes the brain want candy and chocolate, and the gut makes us depressed, and it's really interesting because the gut microbiota does produce a lot of neurochemicals. Hundreds of neurochemicals are produced in the gut. And then information is sent via the vagus nerve to the brain. And the brain then uses these chemistry to regulate basic physiological processes as well as mental processes, such as learning, memory, and mood. For example, gut bacteria produces serotonin and vitamins. And if we're eating the wrong diet, we may deselect for these good bacteria, and we may feel depressed. So the vagus nerve then connects the brain, the nervous system, with the immune system and the endocrine system. And the endocrine system is our hormonal system. And then, of course, the microbiota is supporting hundreds and hundreds of signals for this system to operate. And we really do have an impact on how the system is functioning. We know that there's food out there that makes us feel good. It's called probiotics. And these foods you probably meet every day. It's kefir, cheese, yogurt, sauerkraut, kombucha, miso soup, soy, sourdough. And then, of course, the industry and the health industry has come up with a bunch of other dietary supplements we can purchase in the store. It's a multi-billion industry right now. There's very few evidence, scientific evidence, showing that whatever you can buy in the pharmacy actually has a good impact on your health. But there's at least anecdotal information showing that we do feel good from eating probiotics food. I dove a little bit deeper into this. Today there's a really popular drink called kombucha that is marketed as providing anti-inflammatory and antioxidant properties. And it tastes really good, actually. And it is a symbiotic culture of bacteria and yeast, SCOBY it's called. So you grow bacteria and yeast in sugar and tea, so you can use green tea, you can make this at home if you want in your own kitchen. And then after a few weeks, a lot of organic acids and other beneficial metabolites is supposedly formed in this drink. And I thought, well, I like kombucha, so I should dive into the literature and see, is there any evidence showing that this actually has a good impact on my health? So I found 13 published studies, but no controlled studies involving humans. So they were only reporting on anecdotal information collected between 1925 and 1950 that people were feeling good drinking kombucha. So we really need to research more. On the other hand, sauerkraut, which is symbiotic lactic acid bacteria culture, metabolizing cabbage in vinegar. There were actually 138 published studies, and a handful of them show molecular mechanisms showing that which genes were involved in making us feel good, and they showed which molecules. So it turns out these lactic acid bacteria can produce these D-phenylactic acid, amino acid that is, and that triggers the synthesis of another acid by the human cells, and that is very beneficial for the human immune system. So sauerkraut it is. So I did some more research and looked at other probiotics. So I found studies that had that at least contained one randomized controlled trial, and these probiotics included kefir, sauerkraut, and sourdough bread. So kefir, it turned out, it eradicated the very harmful gut bacterium Helicobacter pylori. Helicobacter pylori is known to cause ulcers and also gastric cancers. So kefir it is. But there were no studies available with controlled trials for kombucha, miso, or kimchi, and hopefully other scientists are focusing on this, because I think this is a very important field of research. And it's important to remember too that the science that has been done usually focus on one population, one ethnicity, maybe in Europe, that these responses from the food might be very different depending on our genetic makeup. So we need to understand more about the individual's response, I think. So in my research group, we are very interested in continuing exploring mechanisms behind probiotics. But something that we're working on very actively is developing new drug therapies to eradicate human infectious disease. And I'm talking about antibiotics, antimicrobials. Currently, 17 million people die per year from bacterial infections. This is worldly, and that's a lot of people. And we're currently using the last generations of antibiotics. So many antibiotics that we treat disease with are a broad spectrum. They kill most of our microbiota, even the good bacteria. There's big interest in developing therapies that only targets the pathogens and leave the rest of the microbiota. So that's our idea, to leave the rest of the microbiota intact. So the antimicrobial resistance starts not only in human medicine, but also in food production. We treat humans and animals in food production with the same antibiotics, and that's a huge problem. So our meat industry, poultry, fish, all in preventative purposes, so animals will not become sick during their growth and during the production, not because they are sick. So we could probably reduce this loss by giving animals probiotics instead, maybe. So what we're selecting for then is antimicrobial resistance, which is then spread to the farmers and also to the consumer via the food. And then we end up having cases at the hospital where patients are not responding to antibiotics. So developing more targeted therapies is very important. So then, since I'm a microbiologist and I work with the human microbiota and all the genes that the microbiota encodes, we were thinking, like, how about if our own microbiota can produce antimicrobials that we can use for our therapeutic needs? So looking into where does the therapeutics come from today, we understand that, microbes from soil, microbes from the ocean, from the coral reefs, they're actually contributing with all the pharmaceuticals, almost all, that we're using today. So if we look at the development of natural products between 1940 and 2014, 70% of all the new antibiotics came from the microbial world. 75% of anti-cancer drugs were antimicrobial derived. 50% of all the new immunosuppressants. And here are some examples. Vancomycin and all these drugs that you can see here have saved thousands, millions of lives. We have penicillin, rifamycin. Doxorubicin is an anti-cancer drug. It was identified in the 60s by an Italian scientist. It's the soil bacterium and actinomyces that produces doxorubicin. And it's still the most potent cancer agent we have today. So there's a lot to mine when it comes to drugs, and looking into our own microbiome could really be worth it. So I promised Rina not to show any pathways, but here it is. Hold on. I owe this to the microbes. Because this molecule, the beautiful structure you see here, don't be afraid. It's called daptomycin. This is a really important molecule. It's one of the only antibiotics that we can treat multi-drug resistant gram-positive bacteria with today. It's one of the few. So here's the pathway. It's not so scary. These are 3 giant genes, GPT A, B, C, and D. They fit together like that. So most of the time, these molecules are encoded by highly structured gene clusters in the bacterial genomes, also in the fungal genomes. So within these genes, you see these little colorful circles. There are catalytic domains, we call them, or enzymes. So what they do, starting in one end and very systematically, they knit together these complex molecules by adding simple building blocks such as amino acids, fatty acids, and it continues in a modular manner all the way to the end of the DPD gene, and it's cyclized by this special enzyme in the end there, and then let go of into the environment. So that's daptomycin for you. So by using machine learning, computational algorithms, researchers, we have not only my lab, but other labs, we have worked on gathering all this information from the human microbiota by pattern finding algorithms, basically. So the pioneers in this field, they detected gene clusters that could encode new antibiotics in all the body habitats. So they found 1,800 in the gut microbiota, about 1,800 in the oral microbiota, a lot less on the skin, but still a lot. 500 around that on the skin. So we have to continue to mine. The more genetic information that become available, we continue to mine. And it turns out that their study was really just scratching on the tip of a very, very big iceberg. There's a lot of diversity in our own microbiota. So we regard this richness as a treasure trove, our own medicine cabinet, that we can explore, hopefully, more about in the future. So in my research laboratory, we have a broad knowledge base. We are biochemists, microbiologists, geneticists, analytical chemists, and we try to communicate. So we're growing microbes in cultures in our lab, and we're developing technologies to access this chemistry. We're developing, for example, tests where we can easily visualize where these compounds are produced, because we can just screen blindly or we can be smart about it. So we can add, for example, a nitrogen compound to the culture and switch its color and become ruby red if we have one type of antibiotics growing in there. So I just wanted to mention a couple of projects just to get your interest. So we were studying one species of bacteria called Streptococcus mutans from the oral cavity, and we decided to focus on gene clusters that were small to start with because it's not so complicated. So the gene cluster here, you see the 9 genes, and we thought, oh, it's so small, we're only going to find one compound. Turns out that this gene cluster alone can produce 6 different compounds that all have different bioactivities and strong activities as well. So this, for example, called mutanocycline, is a structure we have determined in our lab. It's anti-inflammatory. And there's also this rhodorocycline A, B, C. These are highly antimicrobial and very targeted towards certain pathogens. And then we find 2 fatty acids that kill fungal pathogens. And this is only from these 9 genes. And then, yeah, that just tells us that we're on to something really exciting here. And since I'm so interested in this the evolution of these gene clusters and how bacteria exchange genes, it was also pretty intriguing to find that this gene cluster had been transferred from lactic acid producing bacteria in sourdough bread to oral bacteria that now can produce these compounds. Yeah. So another strategy we're working on, I'm wrapping it up here, is called the Trojan horse antibiotics strategy. We have found a molecule we have named rothibactin. It's a siderophore, and the bacteria produce it to scavenge or to capture iron in the environment. Metals are very important for all life. We could not be here without metals. And iron is used by bacteria, fungi, and human cells to obtain energy. So iron can be oxidized, it can be reduced, and during these procedures, bacteria or human cell can harness the energy that is released, and the energy goes to growth or the synthesis of other important metabolites. So we can play around with this molecule now that we have isolated from oral bacteria. So we know it's a very strong chelator of iron. So what we can do, we can modify it. So the structure you can see here, the gray and the red and the blue core of it. So we can add a linker to an antibiotic, and we call this the Trojan horse because the bacteria does not know that it's receiving the antibiotics. It thinks, right, that it's receiving this iron molecule that it really, really likes, but it's going to get a surprise because when it incorporates it into the cell, it's going to die because it's an antibiotics. So you can add multiple antibiotics to this and link it to this siderophore because many times bacteria develop resistance very quickly to one antibiotic. But if you have it linked to another, it's almost impossible that it will be resistant to the second antibiotics as well. And hopefully that will ultimately make it into the cell and we can eradicate pathogens such as Pseudomonas aeruginosa or some other terrible bug. So I'd like to wrap this up with my personal advice on oral care. Because the oral cavity is so important and we forget this. So to keep ourselves healthy, we should try and make sure we brush and floss our teeth after every meal. And this is really something difficult to do, I know, but if you can't do that, at least chew on a gum, a xylitol gum. Xylitol is known to remove biofilms on teeth. Or finish your meal with an apple or with nuts, because these are hardy, fibrous materials that will let go of your biofilms. Don't snack too often, because that will reduce pH in your saliva and you can get cavities. Take advantage of your dental insurance and do all the cleaning procedures you're allowed to. Many people don't do that, but really, when you brush, you can only brush. Let's say many adults have problems with pockets, gum pockets, and those pockets can be deeper than 2 millimeters, 4, 5 is not so uncommon. But at home, when you brush and do your cleaning, you can only reach down to 2 millimeter. If you have deeper than that, you really need to go to the dentist and let them take care of that cleaning. Get an electric toothbrush. They reach much better. And I used to do oil pulling. I don't know if you heard about that, instead of using mouthwash. Mouthwash can be dry out your gums and make your gums irritated. So oil pulling is you basically take coconut oil on a tablespoon, or you can do cold pressed sesame oil, and you take that tablespoon, mix it up with a little bit of water, and swish it around for 20 minutes. And you do that, yes, I know, you better not be busy that time. 20 minutes. Yeah, and it's really good exercise, just like you say. You exercise the muscles in your mouth, and it's sort of like a massage for your gums. It's very important to take care of your gums and massage them. And there's also these little tools you can buy at the pharmacy, and they have a little rubber head like this. And you're supposed to put them in between your teeth and circle around a little bit to massage the gums. And yeah, they are. Yeah, so these are just little advice to you. And that you can do what you want with. And in summary, to wrap up the scientific part of this, so I think we still have some way to go until we have the magic potion for each one of us to prevent and treat disease. But I'm very excited that we have gotten access to some of the natural products and small molecules of the human microbiota, and I hope in the future we get the opportunity to put them through clinical trial. And then I am very excited to report that the human microbiota is the treasure trove for in our medicine cabinet for new antibiotics and anti-inflammatory molecules. And with that, I wanted to thank you very much for coming here today. And thanks to Jill, Rena, and Rebecca for arranging. And also my colleagues at J. Craig Venter Institute, my direct co-workers working together with me on this science is postdocs in my lab, Carla Ranga, Jonathan Baker, Xiaoyu Tang, and lab technician Pablo Arroyo and Sandra Labont. And also I would like to thank our president, Karen Nelson, for her support of my science, and my colleague and collaborator, Dr. Marcelo Freire. And I would like to thank Nini Tran at the School of Dentistry at UCLA. She has collected a lot of clinical samples for us. And my mentors and advisor at UCSD, Dr. Peter Dorstein, Rob Knight, Victor Nisset, and Bradley Moore. And to the NIH for supporting some of my research. And thank you again. If you have questions, of course, you're welcome to ask them. Yes, over here.
We've seen some of the good things for a microbiome. What should we be avoiding? What are some of the worst things we could be ingesting to harm our microbiome? Would it be sugars or?
Yeah, so did everybody hear the question? So what is the worst thing we can ingest? So there's a lot of things we are not supposed to do, like eating a lot of processed food, like sugar, for example, and smoking, alcohol, all those fun things, right? So I think it's all about not snacking too much, and when you snack, make sure you brush your teeth, and if you don't have access to that, rinse out your mouth, and eat a mixed diet, I would suggest, and don't just get stuck in one dietary pattern. I think for overall health and exercise. And, yeah.
And water.
Water is really good. Yeah, it's the best drink ever. Yes.
Is there any special or new toothpaste that you suggest?
Is there any special or new toothpaste I suggest? Is the question.
What do you use?
So I've actually used a toothpaste that you can only buy outside the United States. No, it's different brands in different countries actually, because it contains arginine, the amino acid arginine. So what you do is, because for some reason, there's some business reason here in the US, you're not allowed to sell it here. It's really ridiculous, but it really works. So you can go to Mexico. So some of my colleagues live in Mexico. So I asked them to buy it for me. So arginine does, it stimulates the oral microbiome to produce alkaline, that is molecules with high pH. So instead of getting the low pH that wears on your tooth enamel, you stimulate the microbiome to produce these alkaline molecules such as ammonia, and it works. It's proven. Yeah, you're so welcome. Yes. Any other questions? Yes?
Is it possible to target your Trojan horse mechanism to deleterious bacteria, or is it going to hit all of the bacteria in your body?
Yes. So it's possible to use the Trojan horse strategy to target special bacteria. Yes. So the siderophore structure, you saw, I didn't have time to go into it. It contains protein regions that that certain bacteria can recognize and others can't. So you can definitely learn, for many pathogens really appreciate the siderophores because they can grow very fast when they can internalize this iron. And we can find those patterns easily in the genomes. So we can modify the siderophore to target, for example, Pseudomonas aeruginosa, which is what we're working with in my lab right now. Yeah. Mm-hmm. Yes?
Is your institute, can you answer this question? Is your institute working on the new generation of antibiotics to rescue the fact that the current antibiotics are almost at the end of their lifespan? Are you working on that?
Yes, so that's what we're doing when we're mining the microbiome and finding new molecules, new therapeutics, because many of them are antimicrobial. Yes, it's just that we need to put them through clinical trial, and that takes some few years. So yes, we are working on that. Yes?
First, thank you for a lovely presentation. But also, if I don't have time to slop oil around my mouth for 20 minutes, how effective is using a water pick in comparison?
Water picks are really good if you have gum pockets, for sure. So yeah, I will definitely use a water pick. Mm-hmm. And they...
Do you believe in taking a probiotic every day along with kefir? Or can you substitute the kefir for the probiotic?
Substituting kefir with probiotics or take them together. It's difficult because if you feel good from eating that probiotics, go ahead and take it. But there's also studies showing that some people get sick of some probiotics. So now I think your primary physician these days would not probably recommend you a specific probiotic, or they shouldn't, because there's been recent clinical trials showing that it's very person-specific how we react to those probiotics. Some people get tummy aches and don't feel good, and there's toxin produced. Other people feel good. So it's very varied. But I think old traditional probiotics like kefir or sauerkraut, I think is not going to have a major damage on our microbiota. Maybe one problem they have seen is that if you have a low diversity of gut microbes, which is not the best thing to have, you're supposed to have a lot of different bacteria in your gut, that when you take probiotics, that you actually may add to that low diversity, because then you only are eating maybe 8 or even less probiotics bacteria, so you're even lowering the diversity more by doing that. So eating fibrous diet has shown to enrich for lactic acid producing bacteria that are good. Any fibrous diet, lentils, beans, those things that we love, right? And so I think that's a really good way of picking a diet rich in fiber, and then eating kefir is fine, and sauerkraut. Yes, over there in the back.
So sometimes we brush our teeth with baking soda.
Yeah, brushing teeth with baking soda.
Right.
Mm-hmm.
To get rid of some stains, whatever, make the teeth whiter. Would that impact in any way the pH and the probiotic?
pH impacted by baking soda, sure. pH gets higher by baking soda. The carbonate will be making the pH more neutral, which is good. Question is how long that is maintained then in the mouth. But I think it could be a good start. One thing to consider is also grain size in your toothpaste or in your baking soda, so you don't wear on your tooth enamel too much, because we have very little tooth enamel. It's really a gift, the little tooth enamel we have. So you shouldn't be grinding too much on your teeth with stuff. Yes, over there.
Yogurt, is that of any value in terms of your oral microbiome? And what about the probiotic yogurt versus the normal yogurt?
So what about yogurt? Does that have any value? And is it comparable to probiotics? I think it's about the bacterial numbers. If you have a lot of lactic acid bacteria in the yogurt, more than 10 to the 6, 10 to the 7, I don't know. I mean, it's about the numbers actually. So there was somebody from NIH, I remember they said, well, you have to drink like 6 liters then to have an effect of the kefir. So these are just hypotheticals then when you discuss things, you know. So this is things we need to try and get scientific evidence. But people do feel better from kefir, I know, that people have tummy aches. So, yeah. Yes?
What is the effect of artificial sweeteners on the microbiome?
Mm-hmm. Artificial sweeteners do have an effect on the microbiome. We don't really know what that means, what those effects means, but it definitely changes the gut microbiota and the oral microbiota as well. So I would just do, like with everything, moderation. Stevia is a good option, like a plant-based sugar. Yes, question?
Fluoride mouthwash. What about fluoride mouthwash?
Yes, those. What about fluoride mouthwashes? He asks, and there's very different opinions about fluoride. I'm for fluoride because I've seen cases where in areas of the world where children don't get fluoride, and it looks pretty terrible. So not overuse it. That's not good either. But I think using fluoride when you do your cleaning at the dentist is fine. And I think also if you don't have it in your drinking water for children, I think it could be pretty detrimental. So a little bit of fluoride, yes, because it does protect the tooth enamel a lot. Yeah.
Well, thank you very much. Okay, so from now on, we hope you have happy mouths and sparkling teeth. Just want to take a minute to remind you in December we have our holiday party. There'll be refreshments and musical entertainment. So we hope we'll see you all in December. Thanks a lot.